Welcome everyone of the Cognitive Neuroscience session. And my name is Kasper Kondrukiewicz. Let me introduce you our first guest, Dr. Victoria Gazzolla from Amsterdam. Thank you very much for accepting our invitation. Welcome.
Well, thank you for having me and for inviting me. Actually, it's a great pleasure to be here. And good morning, everyone. I know it's Sunday morning, so I try to help you stay awake. So when I was a student, actually, I was always puzzled by the fact that a lot of scientists of studying more everyday topics were bringing examples from their kids. I said, I'm a student. Why are they kind of have this in mind that kids can bring good examples? And then something happened. I became a parent myself. So here I will go with my example with kids. But the thing is that what I want you to, so it's a short video clip, and I would like really to think a little bit about the word empathy and what it means to you by watching this movie. Here it comes. Okay. So empathy is a big word, and I will talk about it more and more during my talk. But I think that when you watch these movies, you can clearly see what happens. You can clearly see that Julia was starting walking. You could really feel the fact that she was not walking properly like an adult, but she had a lot of uncertainties. And you can, although you are just on a chair, you might even have felt a little bit your bump now when she fell. At the same time, you also understood her happiness in her achievement, basically, and her proudness. So all these feelings and these sensations that these actions carry around. And my main question as a scientist is, what are the neural bases for this intuitive and spontaneous understanding of what goes on in other people? So now we can just go back over about 10 years when the mirror neurons have been discovered. I think that most of you have heard of mirror neurons already, but can you lift your hand if you never heard of them? Great. So now I just want to show you a couple of movies, because mostly you read about it, right? And these are really nice because they make clearly the point. So in the first movie, you will see basically the monkey grasping a piece of food. And in the same time, people were recording, basically, from single cells that were placed in in the premotor cortex. And at that point of that discovery, premotor cortex was just thought to take care of programming your own action, nothing else, basically, okay? So the sh that you heard is the discharge of the neuron, while the monkey takes the food. You will see it again here. So the next movie will also start with the monkey grasping, and then you will see something else. The monkey. And now Leonardo is bringing the food to the mouth, or he's grasping it with the hand. And then again, and the neuron is discharging. So this neuron in the premotor cortex was discharging both when the monkey was performing the action and when the monkey would see the experimenter performing a very similar action. And this is basically the definition of a mirror neurons. Okay? These are neurons that discharge during your own experience of an action and during the observation of a very similar action from somebody made by somebody else. But was this discharge happening for every action? Because if that is the case, it's not really meaningful, right? But the answer is, of course, no. And these responses were very much specific. And of course, for instance, if the monkey would break the peanut, he would have the discharge, and also if the monkey would see somebody else breaking the peanut. And here you have just a rastergram with a half and the neuron discharge. And here you see basically the area under the curve. And if you compare this with the monkey squeezing the duck, and observing the experimenter doing the same, then the response was very much less. And then you had neurons with completely different preferences. So at the point of the discovery, the people thought that one of the function that these neurons could really have is translating basically the actions of others into the representation of that action in the observer. And from there, they basically thought that these neurons had a great potential for explaining like imitation and many other things, probably too many nowadays, but that's how it went. And here I want to show you how we normally study mirror-like responses in humans. Because of course it is nice to say that the monkey is a tool to translate what it observes into its own representation, but we are humans and we are curious about ourselves, and we wanted to know whether it was the same in humans. And of course single-cell recording is problematic, and you can easily know why. And so what we do, we show people movies of this type, like performing everyday actions. Let's see, so okay, and then we compare it basically with movies that should work like this, okay, in which the hand comes in and moves around the object but does not interact with the object. And we show these movies while subject is in the scanner, right, in the functional magnetic resonance scanner. So we can picture basically the brain activity while the subject watches these movies. And we can compare the activity coming from this, while observing this movie, against the activity driven by this type of movie. And we need that to really isolate a little bit the specificity. And basically, what you will see now here is the map of the areas that are active during action observation, specifically for goal-directed action. So we have a lot of areas that make sense, and I will go through that a little bit in a moment. And we also asked our subject to perform in the scanner similar actions. Why? Because one of the kind of important characteristic of mirror neurons was really the fact that they were discharging both during the experience and the observation. So we really have our subjects performing actions in the scanner, like swirling a glass of wine or scooping a bit of soup from a bowl. And then here what you see is the map of areas that are activated when you perform an action. And now what we need to do is basically to compare these two actions, and you see in gray here what is here, and in red what is here, and basically what's left are the voxels that are common to both the observation and the execution of actions, in a similar way to the concept of mirror neurons. Of course it's not the same, because we are speaking of voxels, and voxels contain many neurons, so we cannot know for certain that they contain mirror neurons, but at least they have very much similar properties. And indeed, if you also now go and look at the areas that are involved, we see that the premotor areas are there, and here is the location where the mirror neurons have been found at first in the monkey. This is the premotor area, and then you have the inferior parietal, and then the middle temporal gyrus that is basically similar to the superior temporal circles in terms of function in human. But then, one of the advantages of the fMRI is that you can actually see that there are more areas involved to it. Do they really contain mirror neurons? Well, this is something we need to find out, but the properties is definitely there. And these areas, the primary somatosensory cortex, and it makes a lot of sense, because when we act, we need to activate our somatosensation to really control our movements. And also this area is very well connected with the visual system as well, so it makes also a lot of sense that there might be cells with such properties in there as well. And then we found also more areas there. Nowadays, the question whether mirror neurons exist in humans or not is actually answered, because there have been people recording from single cell in humans, from people that had epilepsy. So this is not a question anymore. The question is still, where can we find them, and what is their function? Okay. If you have questions, you can actually also interrupt me. It's fine with that, if it's fine with you. Okay, but then again, which we could find these properties, what is actually the function? Okay, if we really believe that these neurons or voxels play a role, for instance, in the understanding or in the perception of other people, then we can try to see whether perturbing the activity of these neurons is actually interfering with perception or not. And what we decided to do is another experiment. Okay, and watch this. Which one was the heaviest? The first or the second? The second. So we decided to have such a task. I can show you to you again. See. It's difficult, but the second one is the correct response. So people came in, they went to the fMRI. No, this is not a fMRI yet, sorry. So we showed these two movies, and then the idea was basically, okay, if these areas, like the somatosensory cortex, really plays a role in perception, what happens if we go and perturb that area? And as a perturbation method, we decided to use TMS. Do you guys know what TMS is? Yeah? Okay, basically we used it with a protocol in which we just try to perturb the activity of the area for a long period of time. Okay, it was an offline paradigm. And basically if the perturbation works and this area is involved in perception, then we would really expect an impairment in the task. And this was actually what we found. So basically, people, if you would compare a session in which we gave the TMS, but that was deactivated, basically a sharp condition, if you compare it with the condition in which we have actually disturbed the activity of this area, then we would really see that there was a decrease in the task performance. So from there, we could basically conclude that the primary somatosensory areas plays a role in the perception, at least for weight in this case. But was this like specifically for humans, or was in general that the primary somatosensory area is there to help perceiving everything that is related with weight, or perceiving the weight? So we had another task in which we had a ball. Here. And the task was the same. Tell me which one you consider to be the heaviest. It's a bit more difficult in this example, but sorry?
Left one. Left one.
Left one. Actually, I think it was the right one, but it's okay. And but in this task, we didn't observe any perturbation. Okay, so they suggested that the effect of the contribution of the primary somatosensory was really related to human actions. And then we moved, of course, a little bit backward, because with the TMS, although you're quite specific in your localization, you'll never be sure. And in the posterior apparatus, you also have some somatosensory properties as well. And then again, we did not find any effect. And the same, we moved forward to the primary motor, which is, a lot of people would also consider part to be the part of the shared circuits. We also did not find an effect. But somebody else made the experiment perturbing the premotor cortex, and there as well they observed a reduction in the task performance. So basically, both the primary somatosensory and the ventropremotor cortices contributes to the perception of Alice action. And we ran later on an experiment in the scanner again, and we saw that the activity of these two areas is actually really related to each other, it's connected. And I was now speaking mainly of mirroring, and you will hear of simulation as well. But is it really mirroring? So now I will give you an example. Let's say I will split you in two halves, like from here to there, it's one half, and from here to here, it's another half. Okay? And this half has to close their eyes, and the other one not, and they observe me. And when I tell you, you open them again, okay?